Belinda joined us, although she was sick for the last few days. So I'm really glad, Belinda, that you managed to come and give us this fantastic presentation. Belinda is a senior research fellow in the School of Psychology at the University of New South Wales in Sydney, and deputy director of the Refugee Trauma and Recovery Program. For the last 9 years, she has been conducting research that draws upon neuroscience and psychological science framework to understand biological, social, and cultural mechanisms underlying the impact of refugee trauma and stress. And Belinda and her team have been collaborating with STARS, with my service, and I can tell you the way how research is conducted was really mindful of our clients, their needs, and we got results that are really meaningful for our field, but all our clients experienced, they improved just by being in the project because they felt that they were heard, understood, supported, validated. So Belinda, I want to thank you for that as well.
Thanks, Mariana. That's so nice to hear. Well, thank you so much for inviting me to speak today and share some of the outcomes from the project that Mariana has mentioned. It was a large collaborative project between the university and STARS. And so really, I have to acknowledge the enormous contribution of STARS to enabling this project. And I'll talk a little bit more about that as we go through too. I'm just going to share, check I can share my screen successfully. Now, can you see my slides okay? I can't quite tell, so.
Yes, we can, thanks.
And it's the proper slides, not presenter view?
Yes, I think so.
Okay, yeah. Great. Thank you. Don't want to make that mistake again, which I did at a conference. So thank you so much again. And just to begin, I'd like to acknowledge the traditional owners of the land from which we're all gathering today, and I'm on the land of the Gadigal people, and to acknowledge elders past and present. To begin, I'd actually also like to put the acknowledgements. Not often in talks we put them at the end, but I want to bring them first front, up and centre, and to acknowledge the participants that took part in our studies for their courage, their generosity, and their trust in engaging with us and being willing to enter an MRI scanner to have their brain scanned. It's no easy task to do that. I'd also like to acknowledge STARS I've done so far and our funders, and also the collaborators, both at UNSW, University of Sydney, and the University of Melbourne as well, and various research staff that have engaged with us over the years too. So the outline of my talk today, I wanted to start by talking about why study the brain of refugees at all and why that's important, and then to introduce our project and to share with you the outcomes of 5 studies. And some of these are published and some are just, kind of cutting, nearly hot off the press kind of studies that are still in the process of being, reviewed. And then to talk about some of the limitations and future directions, and hopefully there'll be time for questions at the end as well. So one of the important sort of questions to consider is, why is neuroscience research with refugee populations necessary or important? There have been major advances in the understanding of the development of neural models of trauma and PTSD. And a question that I often get asked by those outside of, not working in refugee mental health is, why can't we just apply these models to refugees? It does seem as though there may be limitations to the current models to being applied to refugees, as they just simply may not be sophisticated enough to account for the complexity of refugee experiences. And first of all, a distinguishing characteristic of the refugee experience is the level of trauma exposure. So refugee trauma exposure is complex, it's cumulative, and it is severe. It often occurs as multiple events over prolonged periods of time and is highly interpersonal, including human rights violations, violence, traumatic loss and deprivation. The trauma often occurs within the context of ongoing insecurity and threat, including persecution, and is not only experienced by the individual, but by families, neighborhoods, and entire communities. Additionally, refugees are forced to leave their homes in search of safety and often take dangerous journeys to do so. On the other side of displacement, refugees are often facing long-term temporary accommodation or having to navigate immigration policies of destination countries that are more often than not designed to deter than to welcome. And post-migration, post-displacement, and even upon resettlement, refugees can face significant ongoing stresses and uncertainties related to practical, logistical, and social issues, including feeling isolated, difficulties finding appropriate employment, and so forth, as well as the stress that comes with adjusting to a new culture and a new way of life, and to building a new life. Many groups in our community experience trauma, such as military personnel or first responders to a high level, or ongoing stress, such as living in poverty. But refugees experience these simultaneously. And these experiences seem to have a compounding effect on risk for psychological distress. So it's highly surprising that meta-analyses show that refugees and displaced populations are 3 to 4 times more at risk of depression or PTSD relative to the general community. We know that there is a direct impact of trauma and displacement trauma on increasing risk for disorders in refugee populations, and elite ecological models of refugee mental health also emphasize the impact of daily stresses on psychological functioning, which can also exacerbate the impact of past trauma on mental health. Most research in the refugee mental health space has focused on the measurement of the prevalence of disorders and, looking at risk factors that might be associated with increasing risk or elevation of psychological difficulties. And there has been relatively fewer or more limited research focusing on the mechanisms that drive these psychological outcomes. And this includes brain mechanisms. So I would argue that clinical neuroscience research with refugees is really critical. First, from the perspective that findings from trauma or other stress-exposed populations may not generalize to refugees due to the severity of their trauma exposure, and the fact that many are experiencing significant contextual stress in an ongoing way, even when they're trying to recover from trauma. Another important factor is that refugees are from diverse cultural backgrounds and hold different worldviews and value systems that might affect the experience of trauma and its underlying mechanisms. And overwhelmingly, trauma research has focused on Western-based populations. And this is another important reason why it might be important to look at, conduct research specifically with refugees. Another important factor is that refugees and torture survivors have a right to rehabilitation under international law. And so it's essential that psychological interventions that support the recovery of mental health and trauma of refugees have a sound scientific evidence base. And so this need has really motivated our project to better understand how refugee trauma and stress impacts on the brain using cutting-edge neuroimaging technology. And so I'd now like to talk and introduce to you the neuroimaging project we have developed, and to explain sort of our project in the context of the first study I'd like to present, that really is looking at the individual impacts of PTSD, trauma exposure, and post-migration stress on the brain. So this project was funded by an ARC linkage grant, and over a period of 4 years, we had over 150 participants with a refugee background who were recruited to take part in a series of fMRI studies. This was really a collaboration, as I said, between the universities and with STARTS. The general sort of process was that participants would be referred to the project either from STARTS or by others community and refugee services. And then participants would come along to a STARTS location and participate in a clinical interview. And we spread this interview over 2 sessions. That enabled us plenty of time to discuss the project with potential participants in order to ensure that they felt comfortable enough and had enough information to provide informed consent to take part. Then about a week or so after the final kind of second clinical interview, participants traveled to Royal North Shore Hospital. We provided transport via taxis for them to travel to Royal North Shore Hospital, where they undertook MRI scanning. And through all of this, we did use professional interpreters when needed as well. And during the MRI scan, participants took part in a number of different studies. Some of them were task-related studies, and others were looking at structural or sort of the functional brain at rest. And I'll talk about some of the findings from some of those different methodologies and paradigms. So the first study I'd like to share today was really motivated by this question of whether current neural models of PTSD were adequate to account for refugee traumatic experiences. The PTSD is predominantly considered a disorder of fear learning, threat reactivity, which is underpinned by dysregulated fear brain systems, including hyperactive amygdala and hypoactive anterior cingulate, gyrus, and ventromedial prefrontal cortex, which fails to regulate or downregulate the amygdala and other threat systems of the brain. Now, the vast majority of PTSD studies have considered the neurocorrelates of post-traumatic stress symptoms, but not so much the impact of trauma as well on the brain, irrespective of psychopathology. And this is an important consideration in refugees, given the cumulative nature of their trauma exposure. Smaller number of studies in non-refugee groups have found evidence that there is a direct effect of chronic trauma exposure on the brain, on fear reactivity in the brain, regardless of PTSD. So we're interested to look at this in refugees. And furthermore, considering the ecological model of refugee mental health, refugees also experiencing very significant stress in the post-migration environment. And this contextual stress might have, and it has been suggested, similar effects on the brain as trauma. So we're interested to look at how these 3 factors and to model these neurally and how they affect fear processing in the brain. And to examine this, we used a face perception task during fMRI scanning using culturally diverse facial stimuli. And this is quite a well-evidenced paradigm or task to engage fear systems of the brain. In this particular study, there was 85 participants who did this paradigm. About 2/3 of the sample were male. The mean age was about 38. The largest cohort were originally from Iran, followed by Iraq and Sri Lanka. But we also had a range of participants from many other countries of origin. It's important to note here that the sample was highly trauma exposed. So the average was a mean of 11 different traumatic event types were experienced by this group, and 40% of the sample would reach the diagnostic criteria of PTSD. But there was a full range of PTSD symptoms. So we decided to look at PTSD from a dimensional perspective rather than from a diagnostic perspective. So to consider the impact of severity of symptoms. So first of all, what we found was very interesting. So we performed a number of multivariate regressions and also some psychophysiological interaction analysis looking at connectivity. And so what we did find, first of all, when controlling for trauma and post-migration stress, there was no significant neurocorrelations with PTSD symptoms in the fear condition. However, we did observe significant correlations with trauma load. So more exposure to trauma was associated with increased activity in regions including the bilateral insular, Rolandic operculum, the pregenial ventral anterior cingulate cortex, and the posterior cingulate cortex. Now, these regions are important for fear processing and also form part of the salience network, which is typically hyperactive in PTSD, but in this case, with a level of trauma exposure. I also conducted hyperconnectivity analyses, and we found that connectivity analysis, and we found hypoconnectivity between the insular and bilateral frontal regions, and between the pregenual anterior cingulate and the left anterior insular right motor regions. So this suggests that trauma exposure affects fear reactivity and erodes connectivity between these regions within the salience and also negative affect networks. And finally, we found that higher levels of post-migration stress were specifically associated with stronger engagement in the fusiform face area. Now, this is a region of the brain critical to processing faces. We also found patterns of hyperconnectivity between the face fusiform area and temporal regions and the cerebellum, suggesting that post-migration stress is associated with perhaps a hypersensitivity to fear facial cues. And this has implications for post-settlement adaptation and in social engagement in interpersonal processing. And the findings from this study suggest that refugee trauma and contextual stress can impact on fear circuitry, irrespective of PTSD symptoms. The next set of questions was starting to think about, well, does the specific type of trauma that refugees have experienced matter? And in particular, we were interested in the effect of torture exposure. Now, torture, unfortunately, is still practiced globally in over 140 countries, and one in 5 conflict-affected people are survivors of torture. And it is the strongest predictor of PTSD in conflict-exposed individuals. Now, there are a number of distinguishing characteristics of torture, but people think that it is characterized by a strong sense of emotion dysregulation, disrupted attachment, loss of control, connection with others, and also a disrupted sense of self. And so it's proposed that the mental health consequences of torture may be multifaceted and also distinct from other trauma. So it's important to have a look at what's going on specifically. What we do know already from existing research is that torture exposure affects the structure of the brain in terms of the thinning of the cortex, in terms of reductions in brain volumes and reduced white matter connectivity. And also functionally, it appears to be differences, particularly in threat-related emotion processing and sensory activation functioning in the brain, and particularly incorporating the prefrontal cortex. There are some methodological issues from some of these past studies. One of the considerations is that it is difficult to understand the specific effect of torture because of the mixed samples and groups that studies have utilized, such as not distinguishing between torture exposure and other types of war-related traumatic event exposure, and also mixing trauma and PTSD together. There's also lots of variations between tasks in terms of methodology, and the majority of studies have also taken more of a regional approach and not really considered sort of how torture might affect the brain in terms of a whole brain network system. And so to consider this, we started to think about, well, how does torture exposure affect the intrinsic functional network connectivity in the brain? And so we're particularly interested in how torture might affect 3 core networks that form the triple network model that has been proposed to understand cognitive and affective disorders, including PTSD. And these networks are the central executive network, the salience network, and the default mode network that's shown here in the diagram. And the aim of the study was to see how torture exposure might modulate functional connectivity within these networks. And it's quite an exploratory study. So our key hypothesis was that there would be differential connectivity within these networks and between these networks in torture survivors compared to a control group comprising of refugees who were trauma exposed but had not experienced torture. So torture was verified in our study via 3 steps. First of all, via the question in the Harvard Trauma Questionnaire, and then through the conduct of a semi-structured interview, on torture experiences, and then post-interview via the interviewer completing the torture survivor checklist to ensure that the participants' experiences wouldn't meet the United Nations definition of torture. So we had a final sample of 99 participants in the study. 37 were survivors of torture, and 62 were not exposed to torture. The groups were significantly different on age, on trauma exposure, that's not including exposure to torture, so irrespective of torture, and also on PTSD symptom severity. So we controlled for these factors in the data analysis. This slide is to just demonstrate the nature of the torture exposure of the participants and to show that it was very significant. So participants were mainly exposed to multiple events or sustained exposure over prolonged periods of time, and were across all of the 7 categories of torture according to the Istanbul Protocol. So to analyze our data, we used a data-driven approach called group spatial independence components analysis. And what this does is to consider what are the active networks in the sample, as opposed to imposing a predefined a priori idea of what a network should comprise. And so from this approach, we identified 25 discrete networks and 8 within our areas of interest. And these are pictured here. Interestingly, there was no network that was identified within the salience network. So there's a lot of detail here, but I'll hone in on some of them that are specific to the findings. So the first finding was that there were significant group differences in the connectivity within the left central executive network. This was the only network that was significantly different between the groups in terms of its connectivity within the network. The torture survivor group showed less spectral power in the low frequency range compared to the non-torture survivor group. What this means is that it's a kind of representative of long distance connectivity within the network itself. And this comprises dorsolateral prefrontal cortical regions as well as the posterior temporal regions. Now, the central executive network is really critical in terms of directing attention to what matters in our environment. It serves goal-directed and problem-solving activities and allows us to focus on what's important. And this finding suggests that the functional integrity of this network is somewhat disrupted in the torture survivor group. We also then looked at how the networks were communicating with each other. And what we found, we used dynamic functional network connectivity, which is a highly powerful and replicable tool for mapping how networks integrate and connect with each other over time, within the time of the resting state scan. And so what we found was, again, a key role for the left central executive network, where the torture survivor group showed more positive connectivity between the left CEN and the anterior ventromedial and the anterior dorsomedial default mode network. Now, positive connectivity means that when one network is active, the other is also active at the same time. And this finding suggests tighter connections between the left lateral brain network, which is the central executive network, responsible for controlling cognitive resources to external events, and more of the medially aligned default mode network, which is the basis of internal self-referential processing, and that really performs the foundation for social and emotional functioning. We also found, though, that the torture survivor group showed more negative coupling between the left central executive network and the lateral frontal network, and between the lateral frontal network and the anterior dorsomedial default mode network. Now, negative connectivity means that when one network is more active, the other is relatively less active in this context. So again, this finding indicates that there are some problems with networks communicating within the lateral prefrontal cortex and between the lateral and the medial brain in torture survivors. So the findings here show that torture survivors are presenting with differential connectivity in the left central executive network, which might drive a functional imbalance between the lateral parts of the brain and the medial networks of the brain. Interestingly, this hyperconnectivity between the central executive network and default mode network has been observed in the dissociative subtype of PTSD. And so we tested this post hoc, but we found that the findings could not be attributed to dissociative symptoms between groups. So this pattern of connectivity suggests that there may be disrupted external and internal processing and integration in the torture survivor group. Findings are somewhat consistent with models that emphasize this shutdown response as an adaptive function to torture exposure, and indeed other types of extreme interpersonal violations. So at the time of the trauma, the torture exposure, or the severe interpersonal violation, it might be relatively adaptive to start suppressing emotional reactivity, suppressing the previously bad memories, and managing the strong arousal in the body, which may serve a coping and survival function. But in the longer term, these sorts of shutdown responses may lead to more longer term withdrawal, emotional numbing and overregulation of emotion, dissociative symptoms, inflexibility, and difficulty engaging interpersonally, which have sort of characteristics which have been observed in torture survivors and are published in clinical literature. Overall, the findings are suggesting that torture exposure has a long-term effect on the core functional architecture of the brain. What we were interested in next is in how torture might affect emotional processing itself, rather than just what's happening in the brain at rest. In this particular study, we extended our face perception task to include happy face processing as a signal of social reward. So in this study, we focused just on male participants, as the majority of torture survivors are male. The groups were equal in PTSD symptoms, so we controlled for age, and we also controlled for differences in post-migration stress, because we were particularly just interested in PTSD symptoms and trauma at this point. So the first key finding was that torture survivors showed reduced activation in the ventral striatum. This, of course, is a key region of the reward system of the brain, but they showed this reduced activation associated with PTSD symptoms, particularly avoidance symptoms. No such association was found for the fear condition. We also found that the torture survivor group showed decreased activity in the left hippocampus in response to both fear and happy faces relative to neutral baseline, with higher levels of trauma exposure compared to the non-torture survivor group. Now, the hippocampus, I feel weird sort of explaining these things to this group, but anyway, just in case, the hippocampus is critical, a region to learning, to memory, and to fear, and it's highly sensitive to trauma and stress. So it's highly surprising to see that it is involved and affected in processing of facial cues in torture survivors. We also did find that there was stronger coupling between the hippocampus and frontal, temporoparietal, and the subcortical regions in the torture survivor group compared to non-torture survivor group. And in particular, this was predicted by avoidance and hyperarousal PTSD symptoms. And this suggests that dysregulation within these hippocampal systems is happening in response to fear faces in particular in torture survivors. We didn't see any differences in connectivity in the happy faces. So the implications, the findings of this particular study is that threat and reward systems are also affected in torture survivors. It's not just about fear processing, and that levels of trauma exposure may have generalizable effects on social cue processing regardless of valence. We're in the process of finalizing the analysis of other paradigms that we've collected to see whether torture affects active cognitive inhibitory processes in the brain and also active emotion regulation processes. So hopefully I'll be able to share those maybe next year. And the second and the last two studies I wanted to share was particularly focusing on the role of post-migration stress and how that might affect brain functioning in refugees. And in particular, thinking about different forms of uncertainty that refugees experience. The first study I wanted to focus on was on refugee visa insecurity. Now, in Australia and increasingly in other countries around the world, many people and many refugees are provided only with temporary visas. This as a policy of migration management and deterrence. So under current Australian immigration policies, arrival by boat or other irregular pathways means that asylum seekers found to be refugees and requiring protection are given forms of temporary visas, sometimes a bridging visa, sometimes a temporary protection or safe haven enterprise visa. So recipients of these visas have rights to work, to live in Australia, have Medicare access, access to schooling and so forth, but have other restrictions, including to family reunification. But one of the key ones is that they are having to reapply for their visa every 6 months to 5 years, and therefore having to reassert their claims for refugee status. And the result of this is a temporary status with a life lived with uncertainty over the future. We know that this uncertainty adversely affects mental health and self-determination. Research has shown that refugees with insecure visas have heightened depression, PTSD symptoms, suicidality, and lower quality of life compared to refugees with secure visas. This heightened sense of uncertainty that is the hallmark of insecure visa status might have neural underpinnings, but no study has really examined this. There is a new model that suggests that uncertainty has a negative emotional effect because it directs mental simulations about alternative possibilities of the future. Mental simulations can include activating the imagination to think about what might happen in the future, mind wandering and daydreaming and so forth. You can imagine that this does have relevance for refugees with insecure visa status, who might imagine a future of having to return to a country where they are persecuted or being taken to an immigration facility, for example. This functionality is really highly dependent on the default mode network of the brain. Now, we talked about the default mode network in terms of being impacted by torture and the connectivity between the central executive network and the default mode network in torture survivors. Now, the default mode network is our most active brain network when we're at rest, and internally it works to promote self-knowledge and understanding. It does consist of a number of subnetworks, and one important one is the anterior prefrontal hub and the posterior hub, which comprises of the posterior cingulate cortex primarily. These midline hubs are very interconnected with one another and highly responsible for self-referential processing. And that is thinking about the self and imagining the self in the past, the present, and the future to promote self-conceptualization. So we thought that maybe if the visa insecurity is affecting the sense of self and a truncated sense of self in the future, we might see differences in refugees with visa insecurity in terms of how the default mode network is functioning compared to refugees with secure visas. And so to do this, we did look at resting state fMRI and compared a sample of insecure visa holders, so 47, to secure visa holders, which were 51 participants. The groups were equal on all other dimensions, apart from age, which we controlled for analyses. I'm realizing the time, so I'm just going to kind of quickly go through some of these, but this is showing that the insecure visa group in blue showed much more increased levels of different types of post-migration stress compared to the insecure visa group. Again, we used the same data-driven approach called group spatial independence components analysis, and we found 10 regions with our networks of interest. So first of all, we found that there was one network where there were significant group differences, and this was the anterior ventral default mode network, which showed less activation compared to in the insecure group compared to the secure group. This may reflect less metabolic activation in this region compared to in this insecure group. We also found, using spatial mapping, decreased activity in the posterior default mode network, and in particular a site within the posterior superior frontal gyrus in the insecure visa group shown here in the blue, reflecting less activation or less connectivity in this region. What was really interesting were the findings when we started looking at how these different networks interacted with one another. And we found only 2 networks where connectivity differed between the insecure group and the secure group. So the secure group showed positive coupling between the anterior hub of the default mode network and a posterior hub of the default mode network. This reflects stronger coupling or more synchronization between these regions, which is actually how these hubs are supposed to function when they generally are coactive with one another. What was very striking is that in the insecure group, we found increased negative connectivity between these same 2 hubs, perhaps reflecting anti-synchronous activation. As one hub increases, the connectivity in the other hub decreases. And furthermore, in post-hoc tests, we found that the degree of connectivity between these networks was negatively correlated with the degree of reported fear of deportation, one of the post-migration stresses specifically related to insecure visa status. These findings are suggesting that refugee visa insecurity affects the connectivity within key midline hubs of the default mode network, which were correlated with self-reported fears of future deportation. And these hubs are supporting self-referential functioning and making representations about the self and the future. Other findings have found that stress and psychopathology are associated with similar patterns of anti-synchronization in the anterior and posterior regions of the default mode network. So an alternative explanation is that this is not about uncertainty associated with visa insecurity, but more about the stress, the enhanced stresses that refugees living with insecure visas are experiencing. But it could represent this dysregulated default mode network in terms of functioning within the midline hubs, could be a neural marker of visa insecurities for refugees. And in the final study today, I wanted to share sort of a different experimental study where we reported on the social impact of another form of uncertainty, and that is family separation, and in the context of attachment. So in this study, we had 50 refugees with and without PTSD, and they completed an attachment priming threat perception task, where participants were presented with an attachment prime cue, which were either consisted of parent-child dyads, or in sort of broader concepts of attachment, such as family-related images, compared to a non-attachment prime, which was a singular person. It was matched on valence, so it was a positive image as well. Following priming, participants then viewed an emotional cue, or as a negative or a neutral cue, and then made a rating based on emotional rating of that cue. We also, in the clinical interview, we took a measurement of not so much. We adapted a prolonged grief measure to understand how much grief participants were experiencing in relation to separation from important family members. And so we created an index of separation grief related to symptoms of yearning, emotional pain connected to that separation. And so just to explain these findings in brief, what was so interesting was that study found that refugees with PTSD who are experiencing more grief related to their separated family members showed greater emotion dysregulation in the brain during attachment priming, including attachment prime threat. So what this suggests is that the attachment systems of refugees with PTSD which normally assists to down-regulate threat reactivity, was less effective in doing so when experiencing more grief related to family separation. This resulted in increased amygdala activation and reduced ventromedial, prefrontal, and hippocampal activity. This perhaps could be some neurobiological evidence that family separation really diminishes these intrinsic attachment stress coping mechanisms in the brain for refugees. And so just to sort of summarize the findings across the studies, the first key finding so far is that cumulative trauma and post-migration stress impact on fear and salience neurocircuitry, irrespective of psychopathology or PTSD psychopathology in refugees. The second set of findings focused on torture and found that torture trauma has a long-term effect on brain functioning and connectivity in terms of intrinsic brain networks and in terms of emotion systems, and particularly including reward circuitry of the brain. We also found that refugee visa insecurity disrupts the default mode network, and that separation grief reduces intrinsic attachment and threat regulation systems of the refugees with PTSD. There are some limitations of the studies that we've done. The sample is largely a treatment-seeking sample and combining with some community participants. There was also a very highly trauma-exposed sample, which is, of course, unavoidable in a sample of refugees. We did not have a control group, a non-refugee control group, to isolate the role of refugee status. The project was specifically designed to look at the effect of torture. So the way we recruited was to recruit torture survivors compared to non-torture survivors, but we don't have a control group specifically to isolate the experience of refugee displacement. We also had a smaller sample of torture survivors compared to non-torture survivors, although the sample size is it's a reasonable sample size for the study that we've done. There's other sort of limitations in terms of treatment. So participants were all receiving their standard treatments when they conducted did the fMRI, which included many being on psychotropic medications. We did not require them to wash out their medication to participate. But one important factor was that treatment was relatively stable prior to doing the MRI. There's various comorbidities that we haven't always accounted for, such as depression and complex PTSD. So, a highly complex psychopathology profile that many people experience. So we've largely focused on PTSD in the interest of trying to understand whether or not the neural pathways are affected in the same way with refugees with PTSD compared to what's generally been reported in the literature. There's other ones there, but one of the other things is that these studies are experimental and it's still correlational, and so causality can't be assumed. In terms of future directions, we have new outcomes relating to other core processes. In terms of looking at, as I mentioned before, I'm very interested in applying some more neurocomputational methods to really starting to tease out some of the various complex contributions and contributors and determinants of brain functioning in refugees. It'd be really interesting to start to think about other ways to combine fMRI with other neurobiological and psychophysiological insights. And of course, we've been focusing much more on sort of a deficit model, but to try to start uncovering what are the factors that contribute to resilience and adaptation despite experiencing these very difficult and challenging events. And then finally, to sort of start thinking about, how can these findings be applied, and to move towards neuroscience-informed intervention and also policy. It's a really interest of mine to think about, how can these findings actually contribute to evidence-based policy development and some of the advocacy efforts in terms of shifting some of these more adversarial policies relating to refugees. And that's the end. Thank you so much.
Thanks, Belinda. Thank you. There is a lot of information here. And I believe that for us as neurofeedback therapists, some of the findings that you mentioned are highly relevant for our work. So it would be great if you can share your slides, because I think we'll need time to process and go back and look and think about that later. So if you send us your slides, then we will distribute to other participants, and probably we'll have a chance later to contact you and have dialogue about what that really means for our work.
Of course.
But what I didn't mention is that Belinda is really one of the big advocates for human rights, for rights of our clients. And with your findings and your research, you're really helping to shift that understanding, or shift that prejudices around refugees and what it means to being exposed to torture and refugee trauma. But also the way how you talk and outside distribute information about impact of torture on brain and impact of refugee trauma on brain helps to lay by people, to the people who are not specialists and scientists, to understand meaning of that. So you were chosen in one of 5 scientists by ABC and awarded in 2018 as a top 5 scientists for your contribution in giving other people, non-scientists, understanding of your research, and then generally research, neuroscience research, and how that can help all of us to improve the way how we work. So that's why we invited you, because I believe that your work has meaning for our field. And I thank you for that. And I thank you for your contribution to STARS and our work at STARS, because already your research, your findings are helping us to shape the way how we understand and how we work with clients, and of course, how we advocate on their behalf.
Thanks, Mariana.
So we have a few minutes before our AGM. Actually, we are 5 minutes behind, but I will allow a few minutes if there are any questions.
Yeah, I've got a question. Belinda, thank you so much for your dedication. It's such an incredible presentation. I recently came across with article, I don't remember, I think it was New York Times, describing the experience of the people in the Ukraine, that they were worst type of trauma. They reckon there was one of the worst type of trauma, because the Russian soldiers were raping Ukrainian women because they were Ukrainians. And I'm just thinking, what's your perspective on that? What do you think? Why do you think it's the worst type of trauma? It's kind of like a genocide. Yeah.
I mean, that's a huge question, but I think it is because it violates your understanding of yourself and the world, and it sort of disrupts sort of your moral fabric. I think that is partly why these sorts of events are so damaging for people and difficult to recover from. Also, if it involves loved ones, that can be particularly difficult as well. But my understanding is that the atrocities in Ukraine at the moment are, very high on the sort of the level of sort of the most sort of severe, human rights violations that can be kind of experienced. So, as I said, that the participants in this study, our studies really had a huge range of experiences. Some were very much on the very severe level, and some were of perhaps of a relatively more moderate experiences. So, it was I think that's one of the next sort of steps, is to try to start looking at some of that variability, and starting to piece together why and how these experiences actually do change people, because I think this has a real important role to play in some of the work in terms of highlighting that these, the human rights elements of these experiences and why these events shouldn't, are against international law, and also to help survivors to understand that, that their experiences and the effects of those experiences aren't, do have a biological basis as well. That that's not just something that they might be, making up to get compensation, or these sorts of things can be really powerful tools to help people in various ways.
Thank you so much. It'd be very interesting to research this particular application for therapy, how to help. Thank you.
Thank you so much.
Thank you.
Thank you, Belinda.
I'm putting the sun in my hands.
Thank you for coming, although you were not feeling well for the last.
Thank you. Just ignore my eye. I'm going to.